So it is a chain of 10 reactions. So totally there are 10 reactions in this pathway. So there are different enzymes that are taking place to produce the pyruvate from the glucose. So while studying the steps of glycolysis, please note that the steps at which utilization or synthesis of ATP or NADH is take place. That is, at which step the NADP and the ATP is used, we have to note it. So first step, the ATP is getting consumed, and the third step, ATP is getting consumed. So till now, so where NADH is also should be noted. So till now we did not encounter any NADH. In future it will be encountered. So ATP is utilized at two steps. So first that we saw now. So the first step and in the second step. Okay. So the fructose 1,6 bisphosphate is split into dihydroxyacetone phosphate and 3-phosphoglyceride. Here in the fourth step, the fructose 1,6 bisphosphate, is getting converted into triose phosphate, that is glyceraldehyde 3-phosphate, as well as another triose phosphate, dihydroxyacetone phosphate. Okay, and what enzyme it is using? Aldolase enzyme. That is, here it is known as a cleavage. Okay, it is used for cleavage. So it is cleaving this phosphate into 3 carbon components. Okay, one is an aldehyde component, that is glyceraldehyde. Another one is a ketone component and dihydroxyacetone phosphate. Okay, this is the fourth step. We find that there is one step where NADH plus H plus is formed from NAD plus. This is when 3-phosphoglyceraldehyde is converted into 1,3 bisphosphoglyceraldehyde BPGA. Okay, so here the triose phosphate glyceraldehyde 3-phosphate is getting converted into 2-triose bisphosphate. So this is the fifth reaction. Okay, this is the fifth reaction. 2-triose bisphosphate.
Go on a break.
That is 1,3 bisphosphoglyceraldehyde. As this is a 3 carbon compound, this is also a 3 carbon compound. Here using the NAD plus, it is getting converted into NADH and H plus. So in the fifth step also it is getting converted. We have to note that. Next is the two redox equivalents are removed in the form of 2 hydrogen atoms from PgAl and transferred to a molecule of NAD+. The PgAl is oxidized and with inorganic phosphate to get converted into BPGA. So here 2 hydrogen molecules is removed and getting attached with its NAD+, and hence it forms 1,3 bisphosphoglyceric acid. Okay,
Start now.
this is BPGA. So the conversion of BPGA to 3-phosphoglyceric acid PGA is also an energy yielding process. This energy is trapped by the formation of ATP. Okay, so see here, from this BPGA, we are using an ADP, adenosine diphosphate, that is a 2-phosphate containing group is getting converted into adenosine triphosphate. So from this, one phosphate is getting added to that of the adenosine triphosphate and getting we are forming an adenosine triphosphate, which is an energy. Okay, so here one ATP is formed. We have to note the step. ATP is formed. So another ATP is synthesized during the conversion of PEP to pyruvic acid. So in the next step, the 3-phosphoglyceric acid, which is a 3 carbon compound, it is getting converted into 2,2-phosphoglycerate, and this 2,2-phosphoglycerate by removing an H2O molecule forms to phosphoenol pyruvate. Okay, and using this, here also an ADP is getting converted into a ATP molecule, that is an energy yielding process, and here this is second energy formed. Okay, and finally by forming an energy, it forms 2 pyruvic acid, which is a 3 carbon compound. Okay, this is the steps involved in the glycolysis process. The pyruvic acid is then the key product of glycolysis. Very, very important. What is the key product of glycolysis? What is the metabolic rate of pyruvate? This depends on the cellular need. So how much a cellular body needs, that much speed a metabolism takes place, and the pyruvic acid is formed as a end product. So there are three major ways in which the different cell handles pyruvic acid. So the formed pyruvic acid is handled in three major ways produced by the glycolysis. These are lactic acid fermentation, alcoholic fermentation, aerobic respiration. So this formed PA, that is pyruvic acid, it utilized in three ways. One is lactic acid fermentation process. Next is alcoholic fermentation process. Third is aerobic respiration. So all these processes use PA, that is pyruvic acid. Okay, the fermentation takes place under anaerobic conditions only. Okay, so the fermentation is a process that takes place in anaerobic conditions in many prokaryotes and unicellular eukaryotes. For the complete oxidation of glucose to CO2 and H2O, however, organisms adopt Krebs cycle, which is also known as aerobic respiration. This requires O2 supply. Okay, so for complete oxidation of glucose, so this glucose to get completely oxidized to CO2 and H2O in the presence of oxygen, okay, is known as Krebs cycle. Krebs cycle. This is an important cycle which uses oxygen supply, and it is taking place only in a aerobic respiration as oxygen is needed. So this is the process of glycolysis, and this pathway, glycolysis, steps involved in glycolysis and the enzyme required for the glycolysis will be uploaded in a separate video. And hope you enjoyed with this video. Any doubts, please let me know in the comment section below. Thank you.